Kazakhstan is a country located in the heart of Eurasia, appeared on the geopolitical map only in 1991. It is bounded on the northwest and north by Russia, on the east by China, and on the south by Kyrgyzstan, Uzbekistan, the Aral Sea and Turkmenistan. The Caspian Sea bounds Kazakhstan to the southwest. Kazakhstan is the largest country in Central Asia and the ninth largest in the world. For centuries, the Great Kazakh Steppe accepted caravans of the Silk Road in oases of its cities and settlements. It was historically closely tied to the Silk Road trade routes, acting as a crossroads for the movement of people, goods and ideas between Europe and Asia. The capital of Kazakhstan and its largest business center is Nur-Sultan. The third megapolis of the country, Shymkent, is as a city in the south of Kazakhstan with an 800 years history, founded in the 12th century. Nowadays, Shymkent is a city of national significance, one of the largest industrial, commercial and cultural centers of the country, the third largest by population of more than 1 million people. One of the large multidisciplinary education institutions of Kazakhstan with a deep history is located here. It is Awezov University. That is, here students and undergraduates acquire practical working skills. In other words, this is a mini copy of a large production and dairy plant. Within the framework of the program of scholarship, academic mobility and double degree education, the university cooperates with more than 170 foreign universities. 150 students a year study for the academic mobility at leading universities in the world. The digitalization process has affected all spheres of society, including education. At Awezov University, innovations are intensively introduced into the education system, and 3D, VR, STEM technologies are widely used. The IT Center provides consulting to teachers of schools in Shymkent on robotics, provides material resources to students in the Laboratory of Robotics and Mechatronics at the Laboratory of Mathematical and 3D Modeling, and provides material resources of the IT Center to students and teachers of the Awezov University. In Student Design Bureau, students and teachers prepare scientific projects for participation in regional, national and international competitions. Students of clubs and Student Design Bureau develop robotic systems for participation in competitions and competitions in robotics. The material and technical base of the university consists of 10 educational buildings of a modern standard, 6 student dormitories and 2 sports complexes. For the comprehensive development of students, more than 30 public, political and cultural organizations function. Awezov University, that has united history and modernity, today prepares more than 27,000 students and invites to take your best.
Hello, my name is Michael Houghton. I work at the University of Alberta in Edmonton, Canada. And today it's my pleasure to join you and to give you my thoughts on how I see biomedicine and molecular medicine evolve over the next several decades. First of all, though, I would like to describe to you the work that I did to identify the hepatitis C virus in the 1980s. Hepatitis C virus became apparent back in 1974. By that time, hepatitis A virus and hepatitis B virus had been discovered, and serologic tests for both of those viruses were available. To everyone's surprise, when those tests were used on cases of post-transfusion hepatitis that occurred after transfusion of multiple donors, multiple donations, it was found that neither hepatitis A nor hepatitis B was responsible for the vast majority of those transfusion hepatitis cases. Hence, the term non-A, non-B hepatitis was formed back in 1974, and it stimulated an intensive search around the world to find the causative agent of non-A, non-B hepatitis. I joined that search in 1982 in laboratories in California at the biotech company called Chiron Corporation. At that point in time, in 1982, I was trying to use molecular biological methods to try to identify hepatitis C. But the toolbox was really quite bare. PCR, polymerase chain reaction to amplify nucleic acid, had not yet been discovered and was not in routine use until the end of the 80s. Nobody had identified a non-A, non-B hepatitis specific antigen or specific antibody. There was no cell culture system for growing the agent of non-A, non-B hepatitis. And of course, in those days, we didn't have the benefit of modern technologies, like deep sequencing, high throughput sequencing. So we had no molecular handle for this virus. What we did have were really the materials. We had human biopsies, liver biopsies from patients. We had blood from patients. And also, various groups had developed a chimpanzee model, enabling transmission of non-A, non-B hepatitis from humans to chimpanzees. And it was known that the infectious titers of non-A, non-B hepatitis were generally much lower than those for hepatitis A and hepatitis B. So my laboratory in California worked on this for 7 years, from 1982 to 1989. And we obviously had a lot of failures for a long time, but eventually we were able to identify hepatitis C virus using a molecular biological approach, which is indicated on this slide. So what we did is to take chimpanzee plasma that was known to be able to transmit non-A, non-B hepatitis to other chimpanzees via inoculation intravenously. Not knowing what kind of virus this was, whether it was big or large, whether it was RNA or DNA, single-stranded or double-stranded, we ultracentrifuged this chimpanzee plasma such that the smallest virus known to man would be pelleted in the bottom of the tube. In other words, we ultracentrifuged it at very high speed, very high G-force for a very long time. We then extracted both RNA and DNA from that pellet because we didn't know what kind of nucleic acid it had. We then converted that into clones into the bacteriophage vector lambda GT11. This is a phage vector that readily infects bacteria and sets up lysogens in such a way that each piece of nucleic acid, each individual piece from this pellet, could actually be cloned into a separate bacterium. And of course, we end up with millions of bacterium, all containing a unique nucleic acid derived from the pelleted chimp plasma. Lambda GT11 is not only an extremely efficient cloning vector. Incidentally, this was developed by scientists at Stanford University in California, but also it expresses the clone nucleic acid into its encoded protein. So essentially, we had several million clones, each expressing individual proteins derived from the chimpanzee plasma. And then the way we identified hepatitis C virus was to take serum from patients diagnosed with non-A, non-B. The antibodies to the virus had not been identified at that point, but we assumed that they were there. And we incubated patient sera with these libraries. You basically blot the libraries onto nitrocellulose filters. You end up with hundreds of filters. You incubate it with diluted serum from patients, and you're looking for positive interactions of an antibody with one of the clones. And we use a radioactivity method to detect the binding of the antibodies. This was a very novel approach to identifying an infectious agent for the first time. And the way that we were able to show that one of these clones, clone 511, was derived from the hepatitis C virus is that, first of all, we showed that it was extra chromosomal. It was not derived from the human genome or the chimpanzee genome. Secondly, we showed that it was derived from a large RNA molecule of about 10,000 nucleotides that was found only in non-A, non-B hepatitis infectious samples. And then thirdly, we showed that 511 encoded a protein that bound antibodies found only in non-A, non-B infected humans and non-A, non-B infected chimpanzees. And then as we sequence more and more of the genome that 511 was derived from, we saw clear, very distant sequence homologies with flaviviruses and togoviruses. So at that point, we knew that we had identified the hepatitis C virus, and we published our work in 1989 after working on it for 7 years. And I say we, the people who contributed to this discovery are shown here. And you can see my colleagues Quilim Chu, Dr. Quilim Chu, Dr. George Kuo, and Dr. Daniel Bradley. Quilim and George worked with me at the biotechnology company we were all working at. And Daniel Bradley was a collaborator from the Centers for Disease Control, and he provided us with the chimpanzee plasma. So very much a team effort. And I think in the future, like the discovery of hepatitis C virus, major medical advances will be done and need to be done by interdisciplinary teams, people that bring some different perspective, some different technology, some different methodology to the problem. So very important for young scientists to collaborate and work together. Well, the long time that it took to identify hepatitis C virus as the major cause of blood-borne non-A, non-B hepatitis, it was around 15 years from 1974 to when we published in Science in 1989. Although it was a long time, once we'd identified the genome, we were able to do a lot of translational work with it. The first thing that we did was to develop diagnostics and blood tests to screen blood donations using antibody tests to detect the presence of Hep C antibodies. Later on, we developed nucleic acid tests as polymerase chain reaction technology was spun out in the late 80s. We were able to develop nucleic acid tests, and the combination of the antibody and the nucleic acid tests essentially stopped the transmission of hepatitis C from blood banks around the world. Secondly, our knowledge of the genomic sequence and structure of the virus led, once again, after a long time, to the development of specific antivirals to inhibit the replication cycle. And these antivirals essentially were shown to cure nearly all HCV patients in a fairly short period of time and by oral administration. This was the product of pharmaceutical companies working with academic centers, working with public health organizations that resulted in a cure for hepatitis C. And thirdly, the identification of the genome of Hep C enabled vaccine strategies to be pursued, and they still are being pursued. So everything in hepatitis C research has taken a very long time, but ultimately we are optimistic that a vaccine can be produced. So that is the history of the discovery of hepatitis C virus. As I mentioned, it was performed in the 80s by my colleagues and myself. It took us 7 years of us working on it, but for the field, it took 15 years. But today, using polymerase chain reaction, deep sequencing technology, and bioinformatics, hepatitis C discovery would probably only take 7 weeks rather than 15 years. And so you as young scientists will participate in the evolution of yet more sophisticated technology, enhancements of deep sequencing, mass spectrometry, bioinformatics, and I see identification of epidemic etiologies, viruses, bacteria, parasites will take days rather than years historically. So these powerful techniques will really enable identification of infectious agents so quickly. Now, the development of direct-acting antivirals against hepatitis C took almost 26 years. We identified the viral genome in 1989. It wasn't until 2014 that Gilead came up with a cocktail of drugs targeting the viral proteins that were able to cure nearly all hepatitis C patients. That was 26 years of dedicated effort from pharmaceutical companies, academic labs, and public health labs. And here is a schematic of the hepatitis C viral genome. It is an RNA genome, as I mentioned. It is classified within the Flaviviridae family as a separate genus from the flaviviruses and from the toga viruses. It's about 10,000 nucleotides. The virus encodes a single long polyprotein from this very large open reading frame shown here. And the polyprotein of around 3,000 amino acids is cleaved during translation and after translation by a combination of host proteases and viral encoded proteases. And so you end up with 3 structural proteins that constitute the components of the virion: the nuclear capsid, which binds to the RNA, as well as the envelope glycoproteins in the virion, and then 2 envelope glycoproteins shown here and here. And so these are the structural proteins that make up the virion. And then all the other proteins shown here are non-structural proteins that are required for replication of the virus. And we and others were able to show that the non-structural protein 5b actually encodes the replicating enzyme, the RNA-dependent polymerase that actually transcribes the genome and replicates the genome. And then we and others were able to show that non-structural protein 3 actually encodes a viral protease that cleaves the polyprotein at these specific junctions, here, 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 and here. These enzymes are absolutely required for the viral life cycle. So many groups developed, inhibitors of the polymerase and the protease. Also, work done by the Rockefeller University by Charlie Rice, as well as in Heidelberg by Ralph Bardenslager, they were able to develop replicons. These are cell lines that are actually replicating the genome. And that work enabled the identification of non-structural protein 5A, an unusual protein, but it was shown in the replicon to be essential for viral secretion from the cells. And drugs, very potent drugs, were developed to this protein by Bristol Myers Squibb, a pharmaceutical company in the USA. And so in 2015, there were two papers published by Dr. Feld and Dr. Foster and their colleagues. They had done phase 3 clinical testing on Gilead's combination of direct-acting antivirals, sofosbuvir, which acts on the polymerase, and velpatasvir, which acts on non-structural protein 5A. Volunteers, obviously hepatitis C patients, were given these two drugs orally every day for 12 weeks. And at the end of therapy, they were interrogated as for the presence of hepatitis C viral RNA in the blood using PCR assays. And the sustained viral response, which means the absence of virus in the blood using PCR, was shown to be nearly 100%, irrespective of the genotype of the virus, whether it was genotype 1, 2, 3, 4, 5, or 6, there was 100% clearance of the virus. And so this was wonderful news that now we could cure hepatitis C patients. We could remove much of the risk to them of developing serious liver disease. But today, we have other tools that have emerged to greatly speed up drug development. And there will be more tools developed over the next 10 years, 20 years. And this will enable very rapid drug discovery and drug development. And this will happen in your careers as molecular medical scientists and doctors. So, for example, up until very recently, if we wanted to identify the structure of an enzyme encoded by hepatitis C, we would have to purify the enzyme to homogeneity. Then we would have to crystallize it, and then we could use X-ray diffraction methodologies to identify the atomic structure. With that information, you could do screening assays looking for small molecule drugs to bind to the protease or the polymerase or NS5A to inhibit its action. And then with the knowledge of the atomic binding mechanism, you can then derive derivatives of more potency. And this is how drug development has been done in the past. But now we don't have to crystallize proteins in order to understand their atomic structure by X-ray diffraction. Now we can use a method called cryo-electron microscopy. We purify the protein, we freeze it at low temperature, and then we can put it into the scanning electron microscope to actually solve the structure straight off. We do not now have to derive crystals. And sometimes it takes a long time to derive good crystals for X-ray diffraction. So this is another method that is being used now to speed up drug discovery. Secondly, we have computationally assisted drug discovery methods to design new drugs. We now can use supercomputers to model the protein structure, and then we can ask the computer to take billions, not millions, but billions of drug fragments that we feed into the computer and ask the computer to figure out if they can bind with high energy. And in this way, we can very rapidly discover new inhibitory drugs. In addition, we can use computation to screen the interaction of our drug inhibitors with off-targets. Usually drugs are specific to the target of choice, but also they tend to bind off target. And this is why we get toxicity usually with drugs. And with computationally assisted drug discovery, we can now figure out what are the off targets and try to minimize those by rational drug design. This is work of my colleague, Dr. Kamlesh Sahu, in our institute at the University of Alberta. And what he is able to do, even without protein structure being available, he can use his computational methods to actually identify inhibitory drugs that the chemists, our chemistry group, can then work on to produce more potent activities. So, for example, if you want to make a drug to protein X, you go to the database to download its sequence. You can then do what's called homology modeling with related proteins where sequence is available and come up with a predicted sequence of your protein of interest. You can then use molecular docking. In the computer, you can dock billions of drug structures to the protein, find out which ones are predicted to bind with the highest energy, and then you can identify a number of different pharmacophores, as they're called, and then you can do further filtering to find out which are the most active drugs in terms of binding to the area of the protein that you're interested in. So this is an emerging science. It is being used now to discover drugs, but this area will enlarge and expand greatly in your careers. This is work that we published in 2014, and there was a hepatitis C drug developed by a pharmaceutical company, and in a clinical trial, unfortunately, it did cause cardiac failure in some of the volunteers. They were all hepatitis C patients. And one of the things we discovered using computational assisted drug discovery is that the drug here, shown in red, could bind to one of the cardiac ion channels, the hERG ion channel. It's a potassium ion channel that allows potassium to get into the heart, and it's one of the essential ion channels to allow the heartbeat. We showed that this drug computationally could bind to this very important cardiac ion channel. And then when we did biological assays to find out if it could inhibit that ion channel, we found, sure enough, it could. And so this is one possible explanation for why these volunteers experience cardiac failure during clinical testing. This is just to give you an idea of what computation can do. Recently, probably many of you have heard about Google DeepMind AlphaFold. This obviously came from Google scientists. And one of the big challenges in the last 50 years is to predict how a protein folds from its primary amino acid sequence. If we could do that with great accuracy, then it would really enhance research, how the protein functions inside the cell or outside of the cell, and secondly, how we can develop drugs to that protein. And so last year, Google DeepMind came up with an AlphaFold algorithm to be able to predict with high accuracy how small proteins can actually fold. And they were able to get a high level of accuracy. And you're seeing schematically, obviously, how this protein sequence was predicted to fold using AlphaFold. So this will be expanded tremendously over the next few years and decades. And so this technology is all converging to greatly enhance our knowledge of protein structure, protein function, biology, and the development of computationally assisted drugs. And so really, I'm quite amazed just how much technology is growing. It's growing exponentially, And you as young scientists, those of you that are passionate about molecular medicine and medical research, you will be part of new frontiers, of identifying new frontiers and pushing our field in a very sophisticated way to come up with new drugs against infectious disease, but also non-infectious disease like cancer. So I think while we're expanding and enlarging our technology capabilities, we mustn't forget the important role of health policy. So public health agencies all over the world play a very important role in protecting citizens of the world. So with hepatitis C, now that we have very good antivirals to cure patients within 2 to 3 months, the World Health Organization, WHO, has recommended a test and treat policy for all countries of the world. So this involves, excuse me, this involves using our blood tests on all the population, or certainly at least part of the population that is high risk for hepatitis C, to find out if they're positive, and when they are positive, treat them with the new drugs to cure them. This will prevent a lot of serious liver disease in the future in these patients, and also will reduce transmission of the virus in the community. And Egypt has really made tremendous progress in testing and treating. And it's something that needs to be done in all countries of the world. In Egypt, they have suffered very much from hepatitis C. Around 7% of the Egyptian population are positive for the virus, which is a very high level. And in rolling out their testing and treating model, Egypt has screened 60 million of their citizens. They found 4 million that were positive for the virus, and then they treated them with the new antivirals, and they cured 4 million people. And this is still ongoing in Egypt. I believe they have 70 or 80 million citizens, and very shortly they will have screened all of their population for hepatitis C and would have treated all of the positive individuals with the drugs and cured them. So this is a wonderful example for what other countries need to do. They need to test all of their populations for hepatitis C. And then when they identify positive carriers, they need to treat them with the new drugs. Also, if we want to eradicate hepatitis C as a major public health problem around the world, we need to develop a vaccine. Vaccines, as many of you know, have been incredibly powerful to protect the human species from a huge array of bacteria, viruses, and parasites. And this will continue to be the case. So we do need to develop a hepatitis C vaccine to develop, to protect people that are susceptible to the virus infection. One of the things that my groups in the US originally and now in Canada have been focusing on is using the 2 envelope glycoproteins, E1 and E2, as vaccine antigens. I'm showing a gel here of E2 shown here and E1 here. These are quite heterogeneous glycoproteins that are on the surface. I should say, excuse me, they are in the lipid envelope of the virus. And these glycoproteins stick out from the surface of the virion and are accessed by antibodies. So these are classic targets for developing protective antibodies and protective vaccines. And what we've done over the years is to test E1 and E2 as preventatives. We've used the chimpanzee model, which is the only immunocompetent, reliable model for human infection. So we vaccinated chimpanzees with E1, E2, and then after vaccination, we challenged them with either homologous or heterologous hepatitis C virus. And what we show down here is that of vaccinated animals, only 16% went on to develop persistent viremia, as opposed to 63% of unimmunized control animals. So this clearly shows that vaccination with E1, E2 can prevent the persistence of virus when the vaccinated individual eventually sees the virus in their bloodstream. And this combination of antigens is the only combination that's been shown to be protective in animal models against hepatitis C. So we've been working very hard on this combination of antigens. We ran a phase 1 trial that was funded by the National Institute of Health in the US, as well as by Chiron Corporation, the company that I worked at, where we discovered the virus. And what I'm showing here is work done in my Alberta lab by my colleague, Dr. John Law, that we published in 2013. We took sera from human volunteers that were willing to be vaccinated with our vaccine. And what I'm showing here is the ability of these sera from vaccinated humans to neutralize the infectivity of different genotypes of the virus. And these are cell-based assays, but each color, if you look at individual number 5, his sera was able to neutralize all of the different genotypes seen around the world, 1A, 1B, 2, 3, 4, 5, 6. So this was a very important finding that we made, that even though the vaccine was derived from a single strain, it has highly conserved neutralizing epitopes, which cause the induction of highly cross-neutralizing antibodies that can inhibit the infectivity of the virus. So this was a very good observation. It gave us a lot of hope that we can develop a vaccine to hepatitis C. And indeed, we are in the process of clinically testing this vaccine itself. Now, while we've been doing this, and once again, it's been taking a very long time, the COVID pandemic taught us that newer technologies using RNA and adenovirus vectored methods can be rolled out very quickly and very effectively against COVID. So we now have new technologies that can be applied to hepatitis C vaccines, as well as any other pathogen vaccine development. And I'm sure you know about the RNA vaccines. Many of us have been immunized with mRNA. Essentially, mRNA encoding the spike protein of the COVID virus is made in the test tube. It's then encapsulated by 4 different lipids, and then we receive the immunization in our arm muscle. And then the lipid particle acts like a virus particle. Our immune system see it as a virus particle. It's presented to antigen-presenting cells, which present the antigen to B cells and T cells, so that we get both antibodies and cellular immune responses. And as you know, these vaccines have been shown to be very effective against COVID. And also work at Oxford University at the Jenner Institute showed that they could use their replication defective adenovirus vectors. They can insert the spike gene into the adenovirus vector, and then after immunization of the deltoid muscle in the arm, the virus gets in. It's a harmless virus. It doesn't replicate, doesn't cause disease, but it does express the spike protein, which then stimulates B cells and T cells and provides very good protection against COVID disease. These vaccines do not completely sterilize. In other words, if you get vaccinated, you can still be infected by the COVID virus. However, and importantly, there's very good protection offered by these vaccines against severe disease that would end up in hospital and obviously can end up with fatalities in unvaccinated people. So everybody should be vaccinated. These days, the mRNA vaccines are preferred. Work has shown that they are safer. But in countries that may not have access to RNA, and many countries didn't for a long time, there was a considerable benefit to taking the adenovirus vaccines, because the risk of COVID disease was much higher than the risk of side effects. But in countries where there is a choice of RNA or adenovirus, countries now choose the mRNA vaccine as efficient and safer. So my conclusions are that powerful technologies are evolving exponentially. And I think this is in such an incredible way that in the careers of the young scientists listening to me that are passionate about molecular medicine and want to enter the field, either as researchers or doctors, you will be responsible and you will take part in what I believe will be a medical renaissance this century. It will lead to the very effective control of both infectious and non-infectious disease, I believe, and I'm convinced of this. So I think any student listening to me that's going to enter biomedicine, you're going to have the most wonderful experience and be extremely effective at helping the human species. I will also emphasize that it's not just molecular biologists, not at all. We need interdisciplinary scientists. We need physicists who are coming up with amazing biophysical and biochemical tools to enable us to understand what proteins are interacting with viral proteins, cellular proteins. And then we need computational scientists who are really responsible for this incredible revolution of computationally assisted drug discovery. And it's only just started. And of course, we have artificial intelligence where we can train our computers, our supercomputers, to recognize, for example, toxic drugs and distinguish them from effective and harmless drugs. So artificial intelligence is going to be very important at very different levels in medicine going forward. And we need highly sophisticated chemists to design better and more effective chemicals, drugs, and quantum computation. In 10 years, it's obvious, I think, that we will have quantum computers. The power of those will be many orders of magnitude greater than our current supercomputers. So just imagine what you will be able to do over the coming decades in your careers. It will be incredible. It will be like magic. And then, of course, public health and policy is going to be very important. So we need all of these medical doctors, physicists, chemists, computational scientists, and public health individuals to work together to really, I think, we will be curing most disease by the end of this century. Well, in ending, I just want to say a few things about the lessons that I think we've learned from the COVID pandemic. And this is work from Dr. Walls and colleagues from the US that were published in Cell in 2020. And